So we've been hearing a lot about exciting experimental therapies that target the underlying DMD genetic mutations, either replacing or correcting those mutations. Yesterday and this morning, we heard about efforts to replace genes. This morning, we are going to talk about gene correction. These are therapies that are designed to restore function to the gene. These target specific underlying genes. And this morning, we're talking about exon skipping and nonsense mutation correction to enable the formulation of functional proteins. So we're way behind schedule, I think, at this point. So we're going to go right into the presentations. We have four: Richard Abel from PTC, Gilmore O'Neill from Sarepta, Michael Panzara from Wave, and Leslie Magnus from NS Pharma. So I would like to get us rolling by introducing Richard Abel.
Thank you. So good morning to everyone, and thank you for the opportunity to be here. My name is Rich Abel. I'm the Executive Director of Global Medical Affairs and the DMD Global Lead for PTC Therapeutics. This morning, I'll provide an update of Adalurin, an investigational dystrophin restoration drug for nonsense mutation DMD. The topics we'll cover today in this presentation is who is PTC, what is Adalurin specifically, layman's term of the mechanism of action, and as well, and what is a nonsense mutation, which is specific for this molecule. I will then show you a snapshot of the extensive amount of work that has gone into the Adalurin clinical development program over the last 20 years. So this is not a new molecule by no stretch of the imagination. And then I'll close basically by reminding you of the ongoing clinical studies that we have. A part of this clinical development program, studies 041, 045, and then 046, and then end with a update of our ongoing observational registry real-world study called STRIDE. This is our forward-looking statement as we are a public company. And we'll start just by giving you a brief history. So who is PTC Therapeutics? It was founded 20 years ago, and since its inception, we as an organization have been passionate about making a difference in the lives of patients with rare disorders. PTC is science-led, global biopharmaceutical company focused on the discovery and development, also the commercialization of clinically differentiated medicines that provide benefits to patients with rare disorders. PTC's ability to bring commercialized products to the market is the foundation that drives this investment of this robust pipeline, which was shown briefly yesterday by one of my colleagues, and to bring also drugs to unmet medical needs. We have multiple programs across genetic disorders, as well as oncology, and if you're interested in seeing this pipeline, please visit our website when you have the opportunity. Yes. Thank you. So PTC Therapeutics, we're the leader in Duchenne muscular dystrophy treatment with 2 of the 3 approved therapies globally, and we have 1 of the 2 here in the United States. These 2 are Adalurin and Inflaza or deflazacort. Adalurin, the first, is the first ever targeted therapy approved for nonsense mutation Duchenne muscular dystrophy anywhere in the world. It was approved by EMA in 2014 and recently by Anvisa for Brazil in April of this year. It's now available in over 40 countries worldwide, ex-US, and we have trials ongoing, which I'll talk about later, which help inform the potential approval here in the United States by 2020. The second is Inflaza, or deflazacort. This is a corticosteroid approved in the US specifically for DMD with both a tablet and a suspension. And if you had the opportunity to see my colleagues talk yesterday during the inflammation presentation late at night, we got out here kind of late, you would agree with this next statement that Emflaza demonstrates best-in-class corticosteroid treatment for DMD. Things happen sometimes quickly, so when I designed these slides, I wanted to say that we were in discussions with EMA around expansion of our label to include non-ambulatory population. Yesterday, we released a press release basically to state that the CHMP of EMA did their review and decided that at this time they would adopt a negative opinion and not expand the label to include non-ambulatory patients. But they did let us know that they would provide a clear update into their EPAR, which is the European Public Assessment report that provides information for the public around a product and why decisions are made for defining the label of certain products or not. And their statement to us is that they're going to clarify to that patients who start Translarna or are already on Translarna who become non-ambulatory while on the therapy do not need to discontinue the therapy. I think this is an amazing win and a step forward for this space and for the patients that we treat currently and plan to go on Translarna soon. Another piece of positive information, so earlier this month for Emflaza specifically, we received an extension for the 2 to 5-year-old patient population for Emflaza by the FDA. So we're proud to say at PTC that we're able to help many thousands of patients with DMD currently with our products and some of the support systems that we have. The remainder of this study will focus on Atalorin. So I'll give you an update of Atalorin. There won't be any new data here today. I want to basically provide you with information about where we are globally and specifically within the US. So this is a conditionally approved product in Europe for ambulatory patients 2 years and older who have nonsense mutation DMD. It's available again to boys in over 40 countries, either commercially or through our PTC Acts, which is an access program for early access or compassionate use in specific countries. We are in active discussions with the FDA regarding approval here in the United States, and we believe that we have a clear pathway forward with very specific requests for clinical data, which we'll go over just briefly in a minute. And for this, also with this in mind, just be mind that this is an investigational product in the United States. I'm watching my time. So in places where Atalorin is approved, it's a small molecule drug that's taken orally 3 times a day at a recommended dose of 40 mg per kilogram per day. It's taken 10 milligrams per kilogram in the morning, 10 in midday, and another 20 milligrams per kilogram in the evening, giving you the 40. We have a clear, robust, I believe, clinical development program, which we'll show in the next few slides. For patients who have nonsense mutations, DMD, and are taking Atalorin versus placebo in these clinical trials, it shows that Atalorin slows the progression of disease, also allows patients to walk longer and stay more physically able. There are fewer accidental falls. It preserves lung functions longer. It improves the quality of life. In addition to this efficacy data, we also believe that it's well tolerated, as the adverse reactions were generally mild to moderate in severity and no treatment-related serious adverse events to report. Based on this, we have, again, positive risk-benefit as determined by the EMA and prompted us the conditional approval in 2014 and then recently by Anvisa. So all of you are aware of the epidemiology and genetics of DMD, so I won't belabor this. There are a few key points that I just want you to take away as it pertains specifically to Atalorin. The incidence of DMD is a range between 1 in 3,600 and 6,000 live male births. This gives about 20,000 children diagnosed with Duchenne globally each year. And it can be caused by the absence or truncated dystrophin, which is caused by a mutation in the dystrophin gene. And what I'm showing you here is a pie graph that is shown in many presentations to show you the distribution of the type of mutations that's possible. And I want to point out here specifically for Adelaurin, it only works in nonsense mutation, which makes up 10 to 15% of the patient population. And then the last piece on this slide is that it's very important that you understand your genotype so that you can receive the right medication. So how does Adelaurin work? In a very simple term, a nonsense mutation in the dystrophin gene again creates a premature termination that forms a shortened or non-functional dystrophin protein. And how does a nonsense mutation work in this space? So a simple analogy that I think is helpful when explaining it to my family members is that if you think about having a conversation with someone and that person places a period in the middle of all of their sentences and they never finish their sentence, then you'll get a sentence that looks similar to this one, where the period or the stop is in the middle. And this sentence will read, "I need coffee in." The rest of the sentence will drop off. And so how does Adelaurin work here? So using a similar analogy, so Adelaurin bypasses the nonsense mutation and allows for the formation of a full-length functional dystrophin protein. And continuing with this analogy, you will be able to read a full sentence, and that sentence will read, "I need coffee in order to function." And one may say that individuals with nonsense mutation, DMD, need Adelaurin in order to generate full-length dystrophin. So we heard earlier a story about life. What life has Adelaurin lived so far? So it was mentioned earlier, PTC has been around for 20 years. It was established in 1998, and Adelaurin was discovered in 2003. Following that 5-year period, an extensive amount of work has gone into Adelaurin clinical development program over the last 15 years. And in that time, we've worked with the community to learn more about the natural history of DMD, which led to the improvement of clinical trial design, and we hope the advancement in early access of, or easier access to approved products for the patient population. We have, in 2014, again, EMA's conditional approval was granted based on the totality of the data at that time, as well as an agreement by PTC that we will continue to have a registry program, which is the STRI 025, which is currently occurring, but also a long-term efficacy and safety study, which I'll talk about in the next slide. And then the other study, the 045 and 046, has been asked for us by the FDA. So these 3 studies are all shown in red here. We have ongoing clinical studies, which I just mentioned. The first is 045. It's a 041, pardon me. It's a long-term study of Adelaurin, placebo-controlled for boys 5 years and older, nonsense mutation DMD. And these patients should be on corticosteroids for at least 12 months and stable for 3 months on the steroid. The study will go for 72 weeks with a blinded 1-to-1, and then another 72 weeks open label. We also have 2 dystrophin studies. So the FDA asked that we provide additional information around Adelaurin's ability to increase dystrophin levels. And so the first study is for naive boys, is 045. We have currently sites recruiting that we believe will be recruited, fully recruited soon. This will look at over a 40-week period. Patients will have samples taken at visit 1, and then 40 weeks later have a second sample taken. There's also 046, which is fully recruited at this time. This one will be for boys or males who have been on Adaluren for at least 9 months. A single biopsy will be taken and levels will be determined. So if you're interested in participating in these studies, at least 041, 045, please confirm dystrophin mutation that you have as a nonsense mutation through full sequencing. Speak to your physician about the inclusion and exclusion criteria of these studies. And then if you need more information, you can reach out to my colleague, Francis Harmon, shown here. The final piece in terms of ongoing study is our STRIDE registry study. So it's a post-approval registry to obtain long-term real-world evidence on the safety and effectiveness of Adaluren. It's an observational study of Adaluren and utilization patterns of routine clinical care. Patients are followed for 5 years. No study medication will be provided, so these are commercially based patients or some who have received it through early access programs can be allowed only in countries where Adaluren is currently available. And just Thursday here, we presented 3 posters of the STRIDE data. I don't know if you had an opportunity to get to it, but just to remind you, the 3 posters, one of which looked at the initial demographics of this patient population with a cutoff date of July 9th, 2018. The safety outcomes were consistent to known safety outcomes reported in our clinical trials. We also reported 1 poster on the long-term efficacy of Adaluren, where we took patients who had a separation of at least 48 weeks from their measurements and several time function tests, and we compared that patient population with the patient population from 020 who received Adaluren or placebo over 48 weeks, and there were 114 in each of those groups. The time function tests that we looked at were 6-minute walk distance, 10-meter run walk, the time to stand from supine, time taken to climb 4 stairs and descend 4 stairs. And these patients showed a reduction in functional decline over 48 weeks, so very positive data overall. And then we also show Kaplan-Meier for the STRIDE patients who were exposed to Adaluren for at least 12 months. There were 207 patient data who were analyzed there, and the average treatment was for 617 days. The mean loss in this patient population was 15.5 years, and as I watch my time, 3? Oh, that's a lot. Okay, so as there's one more piece of positive information, and part of the reason why we didn't have new data here is because we have a late breaker presentation at EAN that's occurring right now, and these data are embargoed until July 2nd when this will be presented. It basically looks at propensity score matched patients from the STRIDE registry where we match them based on time of first symptom, as well as duration on deflazacort and duration on any other corticosteroid. We matched them directly 1-to-1 with patients who are in the natural history database of Synergy. And we are very happy to say that at this meeting we'll provide time of loss of ambulation between these 2 populations. And in the future soon, hopefully, we have some additional data around pulmonary function, which we think that the community will be very excited to see. So with that said, I would like to, again, thank you for this opportunity, but heartfelt thank you to the patients and the families who give us motivation to put our pants on in the morning and get out here and do what we do every day, regardless of our role, and also to those individuals who are responsible for the conduct of these studies, including the investigators, clinical evaluators, and their team. Thank you very much.
Hey, I would like to introduce Gilmore O'Neill with Sarepta.
It's still morning, so I can say good morning. First of all, thanks to Pat and Parent Project Muscular Dystrophy for organizing an annual phenomenal meeting where we jointly reaffirm the urgency of the shared mission that we all have, and also learn from one another how to actually work harder, smarter, and faster to deliver on that mission for everybody. I will actually be discussing scientific hypotheses, clinical hypotheses, and our plans. And as you well know, unfortunately, some of these things never come to pass. And so just understand that what we talk about is looking into the future, and we can never predict the future with absolute certainty. The first thing I will do is affirm the mission for Sarepta, which is to develop life-changing precision genetic medicines to treat 100% of individuals with Duchenne muscular dystrophy. And as a measure of our commitment to that mission, it's important to just highlight our history and where we are today with in excess of a decade of work in Duchenne research, in excess of 500 participants in clinical trials. That includes ongoing and planned participants. And again, heartfelt thanks to all of you and all of those participants for taking on this incredibly important element of the research that we all have to do. And we have approximately 20 clinical trials, both active and planned in the near future. And all of that work is to support and move the development of a pipeline which spans both RNA manipulations, gene therapy, and gene editing. And while you often see this representation of a portfolio, it's important to understand it's nonlinear from the point of view of the timing, because we discover new things as we go along, and so we sometimes slow down or speed up. But I think the important thing for us is that both shared knowledge here and even the shared knowledge we can develop within our portfolio helps us to learn to do things better and faster. So what I'd like to do is focus with the core and preliminary or initial work that we did with our PMO chemistry, which is a phosphorodiamidate morpholino oligonucleotide chemistry, and then show how we are continuing to build on that foundational work. And then ultimately, at the very end, you've already heard about our gene therapy work, address some of the questions around the intersections of these different therapeutic platforms. So obviously, and I think Richard said it very nicely, the fundamental issue in Duchenne is that the dystrophin gene is incapable of making enough or any dystrophin protein to help the critical elements of muscle function. The PMO technology, and in fact the RNA manipulating technologies, use Watson-Crick base pairing to very, very precisely target specific elements of a messenger RNA to block its interaction with splicing machinery. And what does that all mean? Well, the cartoon at the bottom represents a small segment of the dystrophin message RNA spanning exons 46 through, as you'll see in a moment, 58. And I think, again, Richard highlighted or used the reading of a sentence to explain how mutations can interfere with dystrophin production. And in this case, we've highlighted in gray at the bottom an exon 50 deletion. Now, if you think about the disruption to the open reading frame, and you think about the machinery that has to read that frame and then use the instructions on that reading frame to create a protein, you can understand that when we represent that machine with a little car, it drives along till it encounters a deletion and then it can go no further. However, if you skip, in this case, 51 exon, which is downstream of that deletion, the car can continue to read through and it will actually generate a protein off that, what we call truncated message, and include critical elements of the N-terminal, C-terminal, and large elements of the rod domain. Now, using that technology, Sarepta is in a position now in very late development to hopefully help approximately 30% of individuals with Duchenne. Eteplirsen was proved in 2016 for skipping of exon 51. We recently filed a new drug application with the FDA in December of 2018 and anticipate a regulatory action date which is scheduled for August of 2019. And this is to skip exon 53 for Duchenne individuals who are amenable to such therapy. And our casimersen program, which skips exon 45, we are actually looking at the data and have a goal of submitting the NDA in the middle of this year based on findings from intramuscle biopsies from patients in the ESSENCE study. So if we are successful in these filings, then we will have 3 RNA therapies by 2020, hopefully approved in the United States, and obviously we are looking to move beyond the United States. Let me tell you a little bit about the golodirsen program. I think you've heard quite a bit about it, but we just spent a little time here today to discuss it. The program is defined and supported by a series of studies, and the data that I'm going to show you and that has actually been critical to our NDA comes from a dose-ranging and efficacy study done in collaboration with SCIP NMD. And the study 101 is in 2 parts, and that is completed where patients basically, or individuals who participated in the study, went through a dose escalation protocol, and then a 30 milligram dose was taken per milligram per kilogram per week dose was taken forward into part 2 of study 101 with 24 patients aged 6 to 15. And again, important to thank all of you and them for their participation in this study. In addition, the phase 3 study, which is ongoing, the ESSENCE study, 301, and then its extension 302, is actually designed to both look at golodirsen efficacy and casimersen efficacy. Now, let me take you through the data, and I think you're very well aware of what some of the critical questions to answer in any clinical trial would be. First, did exon skipping occur? So we have represented the data here from the 25 individuals in study 101, and if you look at the left hand of the panel on the y-axis, you will see that there's a percentage measure of exon skipping, and then each individual, 1 through 25, is represented at the bottom. At the lower end of the graph, you can see within each individual the gray circles, which represent their baseline muscle biopsy, dystrophin, exon skipping. And that was about a mean of 2.5% at baseline. And then above each gray circle is the corresponding 48-week of post-therapy measurement of exon skipping represented in the purple squares. I think you can appreciate that there was a difference in all patients with a mean increase to 18.9% at week 48. Now, did this associate with dystrophin protein expression? Yes, it did. Again, on the graph on the left-hand side, you will see the mean percent of normal dystrophin expression, and at baseline it was less than 0.1%. And at week 48, we see an absolute increase of in excess of 1% at week 48, representing a 16-fold mean per patient increase in dystrophin compared for baseline to week 48. I think you've already heard as well that trafficking or expression of that protein at the muscle membrane is actually important. We did actually see that, but in the interest of time, I'm not going to show it today. The benefit risk is always an important thing to calculate. And the safety data here are summarized, and we saw adverse events of at least one in all the patients, but the majority of these events were non-serious, mild, and deemed unrelated to study drug. 4 patients experienced serious adverse events, none of which were considered related to study drug. And no serious hypersensitivity events were reported, rash being the most frequent non-serious hypersensitivity event, and no individuals discontinued due to an adverse event. Obviously, the clinical efficacy associated with this is an important question to answer, and the ongoing ESSENCE study will be addressing that. So having talked a little bit about the golodirsen data, which is today's representative of PMO, let me tell you a little more about the PPMO, which is our peptide PMO. And one should take a foundation and build on it. And our desire here is to enhance PMO delivery to the tissues, and we have done that by, or are testing a hypothesis that the conjugation of a cell-penetrant peptide to the PMO chemistry can indeed do that and then associate with improved efficacy. I think you're familiar with the path from the laboratory to the clinic, and I won't belabor that point, but just go straight to talking about some of the non-clinical data in animal studies where we usually use those to do some preliminary dose ranging and safety work. And this is a representation of a single dose of PPMO experiment in MDX mice. Going from left to right, you see increasing doses of the PPMO in that single dose. And the upper panel represents quadriceps or skeletal muscle, and the lower panel represents the heart. And you think you can appreciate going from left to right that you see a change in color from red through a minor degree of yellow to a high degree of yellow. That yellow change represents the superadding of green, which represents dystrophin staining, over red, which is staining for a sarcolemmal protein. And you can actually again appreciate with that dose response that we see for expression and targeting the sarcolemma. To the right are the normal mice, which give you a sort of a reference for comparison. Is this relevant? Well, in the context of 10-week-old MDX mice, we do see a functional benefit associated with that expression. On the left-hand graph, you can see grip strength represented, and you will appreciate that a normal mouse or wild type on the extreme left exerts about 4 femtograms per gram of force. An MDX mouse just treated with a placebo, about less than 3. And then as you go towards the right, you'll appreciate that with increasing doses, we see increasing grip strength, which in fact normalizes in the highest doses. And we see something similar for rotarod, where the measure there is how long it takes for a mouse to drop off a rotating drum owing to fatigue. And that is done safely. The mouse doesn't drop a huge height. But you would again see that a normal mouse will take about 200 seconds to drop. An MDX dystrophin mutant mouse will take approximately 100 seconds. And then you can appreciate again a dose effect in crazily returning that latency to fall to normal. We are in the clinic, and we have completed our single-dose phase 1 study, which was a single ascending dose study, which went up to 6 milligrams per kilogram. And in the interest of time, I won't leave that, but this is all described on clinicaltrials.gov. And then we actually are initiating our 201 study, which is a multiple ascending dose study, which will actually be a global study. But we have already initiated sites here in the United States and will be initiating globally over the coming months. Now, what happens, a big question obviously is what's the next mutation? We're obviously focusing on 51, and I think the key point to make is that we are doubling down on that to ensure we prove the hypothesis regarding efficient delivery of PMO using that conjugated peptide to the muscles. Using that proof of principle in individuals with Duchenne, and ultimately learning the lessons we can, we will then be expanding to other mutations, and I think, or exons, and I think it's important to understand that we do actually want to have a comprehensive approach to that, but we really want to use the 50-51 experiment to understand exactly how we can best do this. And obviously, the science is going to drive that. We have a lot of ongoing activities, as I said, and we are looking at PMO, PPMO, and our gene therapy. And we will have greater clarity probably in the middle of next year to actually make some critical decisions about where we should move our series, our pharmacopoeia of medicines to best help treat everybody with Duchenne. So I did say at the beginning that we talked about gene therapy yesterday, but we should address some of the intersections. In fact, some of those intersections I was going to talk about have already been addressed during yesterday's discussion. But some frequently asked questions are, how do I decide as a parent between exon skipping and gene therapy, or as a person who's actually considering therapy for oneself? And how will exon skipping treatment impact my ability to receive gene therapy later? I think the most important thing to think about when one's considering approved medicines versus gene therapy, which is in development, is the key principle that preserving muscle function as far as one can should be the primary goal. So one should really work very hard with your physician or your child's physician to make sure that you are optimizing the preservation of muscle integrity and function as much as possible. Will exon skipping treatment impact my ability to receive gene therapy or my child's ability to receive gene therapy later? I think that was addressed actually yesterday. I think Lee Sweeney and I think Barry Byrne both commented on that. We do not believe there's any mechanistic overlap between the gene therapy and exon skipping. And probably the best way to just reassure you about that is that the exon skipping sequences would not be able to sterically block or even encounter the target that they're going after in a microdystrophin transgene. I think that's the most important way to put that. And therefore, certainly in the context of should gene therapy be approved, exon skipping prior to that should not be a risk factor or actually preclude use of gene therapy. That's not to say that there might be certain restrictions around clinical trials, but we're talking about therapeutic decision-making, not clinical trials. And so in closing, I just want to say that we have at Sarepta, rather ambitiously built a pipeline dedicated to Duchenne with the goal of treating 100% of individuals with this condition. It is a humbling mission that we've taken on. It's very tough, and it's really good to know that there's so many others who are sharing this mission with us. And I think the most important thing I must do is to thank all of you, very specifically thank all of the individuals who have participated in our trials or are helping us to do a better job of clinical trials, and just ask you to do one thing, which is continue to challenge us to work harder, to work smarter, and to work faster. Thank you very much.
Thank you. And I'd like to introduce Michael Panzara, who represents WAVE.
All right. Well, thank you, Dr. Klein. Thanks to PPMD for this opportunity to speak. It's truly a pleasure to be here to talk about suvodirsen for exon 51 skipping. These are our forward-looking statements, and my disclosure, as was stated, is I'm an employee of WAVE Life Sciences. Before I get into my presentation, I took this timeline from the PPMD website, and I had to acknowledge on this occasion of 25th silver anniversary, it's really been an incredible 25 years under Pat's leadership. The line of progress here is incredible. I've been in other therapeutic areas, and I believe that this is truly unparalleled. And a sincere congratulations to Pat, and the team on this occasion, and on your accomplishments on the behalf of the community and everything you're continuing to do. So thank you very much. This has really been quite a meeting to sort of illustrate that. So as we think about our WAVES approach to genetic medicines, our approach involves oligonucleotide therapeutics. And I think you've heard a lot very nicely described from Gilmore, some of what that means, but there's been 25 years, over the past 25 years, this world of oligonucleotide therapeutics has really been a time of innovation. From the first medicines approved with oligonucleotides in the 1990s, there has just been this continued evolution such that the chemistry, we've advanced approaches to understanding these compounds, and now it's evolved in such a way that the characteristics of these molecules can be precisely controlled, and that's what we call at WAVE PRISM. This is the principle that was behind the founding of WAVE, that this concept, by precisely controlling the chemistry of these oligonucleotides, you can potentially create oligonucleotides that we call stereopure, that have improved characteristics compared with oligonucleotides that have been made in the traditional way, also known as stereorandom. It was the observation by WAVE in 2016 that by controlling the chemistry, particularly stereochemistry, we can improve exon skipping in a dish of cells with deletions amenable to exon 51 skipping that led us to embark on the suvodirsen program, as well as the other exon skipping programs that I'm going to be describing for you today. Now, Gilmore beautifully illustrated exon skipping, but I'm just going to talk a bit about why stereopure oligonucleotides as an approach to treating DMD. Well, first of all is the ability of these molecules to get to muscle cells, both mature cells as well as immature or skeletal cells, or satellite cells. In fact, not only are we finding with these stereopure molecules that they're able to get into the cells, but they're able to get into the nucleus, which is the place in the cell where exon skipping has to occur. And I'll show you some data on that in the coming slides. Next is the ability to administer oligonucleotides repeatedly to address muscle cell turnover in order to accommodate growth of the muscle as the boys grow, and the ability to continue to administer to improve distribution throughout the body so we can get the oligonucleotides to the places where skipping has to occur. Next, this approach also may lead to the production of functional dystrophin that the muscle cells naturally produce. And finally, oligos have been manufactured on wide scale for decades. So even with stereopure oligos, the path to providing enough oligo for medicinal use has been established. It's for this reason that in WAVE, we're really excited about how to approach DMD using the stereopure approach. This slide illustrates one of those advantages. This demonstrates the ability of a stereopure oligonucleotide to get into the nucleus of the cell, which, as I mentioned, is where skipping occurs. This is an experiment where we took cells derived from patients, we put these cells in a petri dish, and we treated with either a stereopure exon skipping molecule like suvodirsen or a stereorandom molecule that targets the same place in the gene. We then looked at the cells under a microscope and we counted the number of red dots, and these red dots illustrate the location of the oligonucleotides in the cells. And you can see that on the right where you have the red dots inside this blue nucleus. We then plotted the number of dots on the graph you're seeing on the left, and we plotted that for both the stereopure molecules as well as the stereorandom. So as you can see, there were a lot of red dots in the nucleus of cells treated with the stereopure oligonucleotide versus stereorandom without a transfection agent. These were just treated naturally, indicating better entry into the cell of the stereopure design. So why is this important? Well, we believe it's important because the more oligo you can get to the nucleus, the more likely that skipping is going to occur. So what you're seeing up on the screen now is a Western blot. This is the way we were measuring the levels of protein produced. In this case, on this Western blot, we were looking at for suvodirsen protein produced by the same types of cells in a dish from a patient who had an amenable deletion to exon 51 skipping, and this was after treatment with suvodirsen in this dish, or the other oligonucleotides. And you can see them there as a PS-ASO, which is a drisapersen analog, a PMO-ASO, or suvodirsen. These are vastly different chemistries, so we wanted to allow them to do this comparison between the three. The green bars near the top of the gel are dystrophin protein. Brighter bars mean more protein. We then compared the amount of protein in these lanes to the level of protein in normal muscle, and that's the bar you see all the way over to the right. The suvodirsen lane is highlighted by the white box. What this shows is that suvodirsen and these other two-wave stereopure oligos produce very high levels of dystrophin relative to the other stereorandom oligos of different chemistries. The numbers represent the percentage of protein versus what is seen in normal muscle, and you can see that underneath. So in other words, suvodirsen-treated cells lead to protein production of about 52% of what you'd see in a normal muscle versus 1% of the other oligonucleotides. Once again, this made us really excited that this was the first indication that we had that suvodirsen might lead to higher levels of skipping and dystrophin expression than had been seen to date, and led us to move forward in the suvodirsen development program, which is really the focus of the rest of the presentation. So this slide shows the overview of the suvodirsen clinical program. As you can see, it's pretty comprehensive. It consists of a multi-center global phase 1 study that was just completed, and the data recently presented at the MDA meeting in April and this past Thursday at this meeting. The open label extension study for patients who participated in the phase 1 study is shown in the middle, and this is where we expect to have data from muscle biopsies taken in this study in the second half of this year. And finally, on the right, you have the Distance 51 study. This is our global multi-center phase 2/3 study to evaluate the efficacy and safety of suvodirsen, namely suvodirsen's ability to both restore dystrophin expression as well as to improve function. The exciting news about the Distance 51 study is that we announced this week that the study is underway, and we intend to begin enrolling soon throughout the world, including the US and Europe. So I'm now going to walk through each of these studies individually, beginning with the phase 1 study, which we found, as presented previously, that suvodirsen was generally safe and well tolerated at doses up to and including 5 milligrams per kilogram. The most common adverse events that we saw in that study were associated with infusions, and we defined that as any event happening within 24 hours after the start of infusion. These were things such as fever, headache, a few episodes of vomiting, a few episodes of a rapid heart rate. These were mild to moderate intensity, and they were resolved with symptomatic treatment being transient over a few hours. We saw similar symptoms with increased severity at doses above 5 milligrams per kilogram. Now, what we learned from this study, perhaps most importantly, is that the concentrations of suvodirsen that we achieved in the blood of patients who received suvodirsen at these dose levels was that which is predicted to lead to skipping. And we learned that both from our in vitro Western blot data, as well as the in vivo studies we've done in animals with this and similar oligonucleotides. So once patients finished the phase 1 study, they were eligible to receive weekly suvodirsen in an open label extension study, or an OLE. Patients first began to enter this study in August of 2018, receiving whatever dose of suvodirsen that they received in the phase 1 study after collection of a muscle biopsy at baseline. Patients continued to receive these doses until we had the phase 1 data and began to transition all of the patients to one of the doses that we had selected for the phase 2/3 study, where we anticipate the greatest likelihood of efficacy. As of a little over a week ago, on January 18th, 25 patients had received at least one of these 2 doses, with several patients already having received 10 or more infusions. and as of that date, a total of 148 doses had been administered. In total, we expect 37 patients to transition from the phase 1 study to the OLE study at one of these doses. The good news here is that patients continue to tolerate suvodirsen well. In fact, the tolerability appears to improve with continued infusions, and that's what I'm going to show you now on this slide. So what you're looking at here is a graph of the proportion of patients with any symptoms within 24 hours after the start of infusion. You have the vertical axis, which shows the proportion of patients, and the horizontal axis, which has the number of doses given, as well as the number of patients who received that number of doses. We also had no discontinuations to date in any of the patients receiving these 2 doses, because what is quite clear here is that there appears to be a sharp decline in infusion-associated symptoms with continued dosing. And every week, this number of patients continues to grow, and again, with the expectation that 37 patients in all will transition to one of the doses in this study. Also, regarding the OLE, we are eagerly awaiting the measurements, the dystrophin measurements, and the muscle biopsies, which we should have in the second half of this year. So now moving on to the last part of my presentation, which will focus on the Distance 51 study. This is, again, our phase 2/3 efficacy study that we just announced had initiated. This is an overview of the study design. This is a global, multi-center study anticipated to enroll in the US, Canada, Europe, Australia, and Japan. Up to 150 boys aged 5 to 12 with mutations amenable to exon 51 skipping will be randomized to one of the 2 doses of suvodirsen or weekly IV placebo for 48 weeks. After those 48 weeks, all patients will transition to the open label phase of the study, where all patients will receive suvodirsen for an additional 48 weeks. So Distance 51 is designed to look at both dystrophin production as well as functional outcomes as shown here, including the North Star Ambulatory Assessment, or the NSAA. In the US and certain other regions, the primary endpoint is change from baseline in dystrophin expression as measured by Western blot. In the EU and Japan, it is change from baseline in the NSAA. The key secondary endpoint in each of these regions is the reverse. In other words, the NSAA in the US or the dystrophin expression in the EU and Japan. We will also be looking at several different measures of strength and function, including the Actimyo, using the Actimyo device, looking at stride velocity, as well as assessments of patient and family quality of life. So one of the many exciting things of the Distance 51 study was its acceptance into the FDA's Complex and Innovative Design Pilot Program, or the CID pilot program. The stated objectives of this program are shown here: to modernize clinical trial design, streamline and advance drug development, and inform regulatory decision-making. In order to get into the program, we had to submit an application that demonstrated that our trial had innovative features, innovative trial design features, as well as was being conducted in an area of high unmet need where no or limited treatment options are available. What we submitted was viewed as innovative because we proposed innovative statistical methods to enable use of historical controlled data, meaning data from the placebo groups of previous trials, as well as other research studies and registries that had been conducted to augment the placebo group. So why did we do that? What does that mean? Basically, it was because as we were preparing for this trial, you provided an enormous amount of feedback that we should try to come up with ways of minimizing placebo exposure, minimizing the time that patients need to be on placebo. And this is what we came up with. We learned that there were many trials, numerous trials, and natural history studies that have been conducted that this community has supported over the years and continues to support that actually recruited the exact types of patients that we are looking for in this study. So we asked ourselves, how do we get those data? Why can't we use those data to try and supplement our study and hopefully reduce the number of placebo patients? And I have to say, once we decided to do that, there were a lot of questions on would we ever be able to do this? And those who came to our support were this community, but also the companies working in this space. Specifically, I have to give recognition to Pfizer, who was very open to giving us placebo patients. PTC stepped up. We have access to their placebo studies. I mean, I think this community owes them, I have to recognize them publicly, it owes them an enormous amount of thanks for opening these data sets to us to hopefully contribute here and contribute to the community overall. So thank you to all of you publicly, also some of the academics who have provided us with these data. And therefore, with these, because of this application, we've met with FDA several times over the past several months to refine the study plan and the analysis with the goal of reducing the number of placebo patients to deliver conclusive results in this study and the number hopefully required so that we can accelerate the development. So more to come on this as we move forward. So I mentioned that our CID program, our entrance into that, was driven primarily by our interactions with the community focused on placebo, but this was really not the only way that you have all contributed to this. First of all, another important thing, we were asked to minimize biopsies as much as possible, but still enable the possibility of accelerated approval or an accelerated regulatory path. We have done this. We were asked to provide and continue to provide access to open label treatment for the boys who've participated in our trials and will participate in the studies, and this is part of the plan. We've also committed to listen and communicate appropriately about our trials and their results. This process began with the presentation of the complete details of our phase 1 study shortly after completion in the appropriate scientific venues and in an open letter to the community, and that is how we'll do things going forward. We've committed to sharing the CID trial design elements as well as the placebo data from this study, just like our partners have, and share those with the community to help stimulate innovation in future clinical trials. And finally, we continue to work with global advocacy groups on study awareness and participant support to enable those patients who may want to enter one of these studies to find a place where they can do that and to ensure once they're in that study, they have the support needed to continue and successfully finish that effort. Finally, this is just the beginning for us with Exon 51. Our Exon 53 program is on track to enter the clinic to enable a data readout next year. And we have numerous exon skipping programs at various preclinical stages, all built upon our learnings from the Exon 51 program. So as a final note, and personally, this has been an extraordinarily fulfilling process for me and for all of the WAVE team. The partnership that we've seen throughout this with patients, with all of you families, people who've participated in our trials, the healthcare providers, advocacy organizations, and our colleagues at the regulatory authorities has truly been extraordinary, and I want to thank all of you again. We at WAVE look forward to continued partnership as we move Distance 51 forward and then develop additional treatments for those people who are really waiting for those big innovations. Thank you very much.
Thank you. And next, I'd like to introduce Leslie Magnus from NS Pharma.
Afternoon, everyone. Thank you for all being such hearty souls and sticking through this whole morning presentation. I know I'm the one wrapping it up, so I hope to go through this quickly and present some interesting information for you. I joined NS Pharma just 2.5 months ago, so you can imagine that the last 2.5 months have been very busy for me, a lot of learning, a lot of information assimilation, and it couldn't have come at a better time because I'm here at my first PPMD conference where I've had the opportunity to not only be informed and learn a lot, but also to get an excellent perspective from the patient and the caregiver directly, which is not something that typically happens in many therapeutic areas. But I'm also glad that I'm here because I have the opportunity to share with you some information about NS Pharma and also about our exon 53 skipping agent, Viltolarsen, which I'm not sure how many people are familiar with. Our goal is to be here at PPMD 1 year from now with an approved product that patients will have available to them. So let me share our forward-looking statement as well. I'll just put that up for a minute. And then let me tell you a little bit about NS Pharma so you can learn a little bit more about us. NS Pharma is a small company. It's based in beautiful Paramus, New Jersey, just a stone's throw from the Garden State Parkway. It's a subsidiary of a Japanese company, Nippon Shinyaku, and we at NS Pharma have been the sponsors and the overseers of the North American Phase 2 trial for Viltolarsen. Our focus area here in the US is on orphan diseases, and with the approval of Viltolarsen, that will represent our first product to market in the US. However, it's important to know that we are an affiliate, as I said, of a Japanese company, Nippon Shinyaku, which is based and has its headquarters in the old capital city of Kyoto, Japan. It was founded in 1919, and on October 1st this year, it will be celebrating its 100th year anniversary. So although we may be a very new company here in the US, we have a lot of heritage and history behind us in our Japanese parent company. Nippon Shinyaku focuses not only on rare diseases and will be hopefully launching Viltolarsen at approximately the same amount of time, or in the same time frame as the US launch. They also have other areas of focus, which include urological diseases, hematology, and gynecology. So again, as you can see, it's a small US company, but we have the support behind us of a larger, older, well-established company in Japan. So many people have taken you through how exon skipping takes place, and I will try to go through this very quickly to move things along. But we are developing a drug for skipping of exon 53, which is designed with exon skipping to create or to fix a problem with a defective coded protein. So how is a normal protein made? Well, I'll just put them all up. Inside the cell, inside the muscle cell in the case of Duchenne, inside the nucleus, you have DNA that's coding for the protein dystrophin. DNA is copied or transcribed into pre-messenger RNA, which is then spliced and diced and forms the final messenger RNA, which then leaves the nucleus of the cell, goes into the cytoplasm or the main body of the cell, and is translated into the functional protein. And that whole process is known as transcription and translation. That final protein that's produced that goes about doing its business. In the case of dystrophin, it's providing that shock absorption feature that we were talking about earlier. But proteins can be other things. They can be antibodies, they can be enzymes. So proteins are ubiquitous throughout the body. In the case of this specific genetic defect, as you can see on the top row, you have the various exons, and as you'll remember, the DMD gene has 79 exons, the largest gene in the human body. In this particular example, you'll see that 48 through 52 are missing, and as the body is assembling its pre-messenger RNA and ultimately its final messenger RNA, it can't code all the way through. It can't be translated all the way through into a normal protein, and thus you have the formation of an abnormal protein. With exon 53, We are deliberately skipping another exon and allowing for the fact, or using the body's ability to connect 47 through 54 in this case, so that when the messenger RNA is being translated into the protein, it is able to read through and form a shorter but still functional protein. And that's why we call it an internally truncated. So the two ends of it that are most critical are now able to be produced. The center portion is a little smaller, or again, truncated. So if we just take it back into the cell, and you can see here in the gene, we have a mutation. The pre-messenger RNA that forms would have a defect or a skip that, when spliced, would form an abnormal messenger RNA. However, by deleting that exon 53, you can now splice the messenger RNA together to form a slightly shorter, but nonetheless somewhat functional protein. And that's the whole concept behind exon skipping, and for our particular medication, Viltolarsen, exon 53 skipping. Viltolarsen was discovered by the parent company, Nippon Shinyaku, and it was discovered jointly with the Japanese National Center of Neurology and Psychiatry, NCNP. It's an antisense oligonucleotide. It has a morpholino background, and it's a neutrally charged molecule. It's a PMO. It's demonstrated exon 53 skipping activity, and it's also demonstrated dystrophin production across several models, including MDX mice. Based on those preclinical findings, it was determined that it should be brought into man, and so clinical trials were initiated. The molecule is given intravenously. It's an IV administration of about 1 hour, and it's given over once a week. The clinical program, as I said, was broken into 2 parts. NS Pharma, the US subsidiary, had a role in 2 of the phase 2 studies. The first was a dose-finding study. It used 2 different strengths of Viltolarsen, 40 milligrams per kilogram and 80 milligrams per kilogram. It was given for 24 weeks. All patients who participated in that study, and there were 16 of them, were allowed and given the option to roll over into an extension study that is still going on to this point. Whoever received 40 milligrams remained on the 40 milligrams. Whoever was receiving 80 milligrams remained on the 80 milligrams. And as I said, all 16 chose to continue receiving Viltolarsen, and at this point, all 16 are still on therapy. Our parent company, Nippon Shinyaku in Japan, launched an investigator-initiated study that kicked this whole clinical process off, but there were lower doses used. You can see here is 1.25, 5, and 20 milligrams. Information from that study is what helped direct us to employ higher doses in our clinical trials. And so Japan launched a phase 1/2 study, which was also dose-finding, using the same doses used in the US, 40 and 80 milligrams for 24 weeks. We are also at the point of discussions with the FDA for embarking upon a phase 3 study to help supplement the information that we're learning from our phase 2 studies. Phase 3 studies typically have larger numbers of patients, and they last longer than the phase 1/2 studies, so these would be longer than the 24 weeks. We are planning to start the phase 3 study sometime this fall. As I said, we are still in discussions with the FDA in determining what the final protocol would look like, but our goal is definitely to have it started by this fall. And when we have finalized that protocol, all details will be posted to, of course, clinicaltrials.gov. With regards to where we stand with Viltolarsen right now, we initiated a rolling NDA submission. The NS Pharma and Nippon Shinyaku plan to complete the NDA submission, as we stated publicly, in September of this year, so September 2019, and we are still on target for that goal. It has been granted rare pediatric disease designation, orphan drug designation, fast track designation, and we are expecting we will get an accelerated approval process. So given all that, we hope to have, as I said, to be standing here at PPMD next year with an approved process. We also have some other exon skipping programs coming up the pike following exon 53. They're still in preclinical stages, but we hope to bring the first one of those into man by next year. So not just one exon skipping agent, but certainly another one following closely behind. And then I would just like to take a moment to acknowledge and thank our participants in the North American sites. We had 5 in the US and 1 in Canada. We also had our Japanese sites and investigators who participated. And then, of course, a big thanks to all our patients who contributed their information and their time to the conduction of this study.
Thank you. Seems like we're catching up on time. So if there are questions from the audience and people want to come to the microphone, that would be great. I have a quick question that I wanted to pose, and I think you've already alluded to it in a way. But for the companies who are doing exon skipping, whether there are plans to roll out follow-up exons and what those plans are and when you expect that to happen.
I mean, I'll be quick because my slide, I mentioned that our exon 53 is entering the clinic such that we expect to have data on that next year, and the other preclinical programs are moving forward behind it, all based on our learnings from 51.
So I also alluded to in the slide, I think we actually have a lot of non-clinical data that we've looked at, we're looking at or have developed across multiple exons. I think the big decision we're looking at is between the PPMO, PMO, and gene therapy, how we're actually combining that. So as I said, we are making decisions in the middle of next year.
The next, is this on? Can people hear me? Okay, it's on, yes. Good, thank you. The next molecule that we're looking at, we have it on our on our page from the parent company, Nippon Shinyaku, if you care to visit that webpage. The next compound up is an exon 44 skipping agent that, as I said, we hope to start in man by next year.
Great. And questions starting on stage right?
Yes. Thank you. I appreciate your talks, and I appreciate your question because it leads to mine. I'm curious why the focus is on skipping single exons at a time, and why not try to skip, say, 45 to 55, like we heard talks this morning with a CRISPR strategy, but using that same type of strategy with exon skipping and pick up, say, 50 to 60% of the entire patient population.
So actually, thanks for that question. It's actually a very interesting one, and actually sometimes it comes down to limitation of technology. I think the way to approach that would actually require probably combining exon skipping molecules. The question then is, one of the things you probably appreciate from today's talk is that what we are trying as the companies are different approaches to enhancing delivery of those targeting molecules, because if you want to combine to actually do what you're describing, you're going to have to hit two. You're going to have to hit simultaneously. You're going to have to bind 2 molecules to the same message to do that. And you actually saw that with a very, very carefully designed CRISPR technology going after the genome, even that efficiency is limited right now. So it really comes down to some of the technological limitations. Does that make sense?
Yeah.
Yeah. No, I would just reiterate that, and I think, as we're learning more and more about our stereopure chemistry, we're trying to understand whether we can get the efficiency to levels to reduce the number of maybe multiple oligos you might need to use in those types of situations.
How's it going? My name is Michael. I'm the proud dad of Mikey and Reed, and Michael is one of the lucky 16 guys who's in your NS Pharma trial. And 2 years ago, I guess when the trial launched, my younger guy, Reed, he was only, I guess, 3, so he's 5 now. And NS, I guess, has kind of hit a roadblock with kind of lining up the sibling dosing. So we've heard from many of the sponsors throughout this, and I can't thank Pat and PPMD enough to allow us to kind of voice our opinions. So I guess I have a weighted question in 2.5 months of being on the job. Here you go. So I was basically wondering, there's so many other families. It seems like a personal question because I am kind of begging to get my other son in. But the parents here, we go through a lot when we try to discover what trial to put our children in, and the amount of, the process that goes through our head is pretty big. And one of the questions that we kind of look at is, are there going to be sibling dosing and things like that? So I guess my big question is, why no sibling dosing for NS Pharma?
I don't know the history behind that here at NS Pharma, but as you said, just being on the job for 2.5 months, it's something that has been raised to my attention, and it's something we have to have internal discussions about and see where we go next. As I said, there's also a phase 3 trial that's starting with U.S. sites. It's actually going to be a global trial, unlike the phase 2 studies.
Do you have the inclusions on that by any chance, like the ages?
I don't have it finalized, but I would assume it would be roughly starting at around 4 as well, which is what the
That's what the current one is, yes.
Study, right. So, and your son is presently 3, or he will be 4?
He's 5 next week.
Oh, okay.
Yeah. So for the past year, he's been eligible for the sibling, but unfortunately, you guys don't have it.
Right. So, but that study will be starting within the next several months. Our hope is to have the U.S. sites up and running first before our other global sites, because we already have that history with the sites that have been participating in our phase 2 study.
Awesome. Thank you.
Yeah, we wouldn't get approval before the studies start, which is, at this moment, we're still in discussions with it, but it may, yes, as we had the whole discussion yesterday. Correct, but the study will be starting before the approval takes place.
Should we move to stage right?
Yeah, my name is Chris, and my son is Henry. He's also one of the lucky 16 that is in the NS Pharma trial. And I was speaking with Terry, so I may have just missed this response, or maybe Pat just asked that. But what dose are you seeking approval on, the 40 milligram or the 80 milligram? And then my other question is, too, going through your presentation, I know there was a little bit of dystrophin data released earlier in the year. Mm-hmm. Why no physical data, the functional testing? How come none of that has been released? Or, I mean, we're 2.5 years into the trial now, and you're in a rolling NDA submission, but we still have no idea what that looks like.
Okay, well, I'm happy to answer that question for you. With regards to the first part of the question, the 40 or 80, that will be decided when we have input from FDA. We'll go to them with all our data, and it's always a negotiation process between what FDA thinks should be the dose and what we think should be the dose, and then a decision will be made. So probably not come out with both doses, but probably pick one, and a lot of that will depend on negotiations with the FDA and how they view the data. With regards to the functional information, that data was presented at World Muscle Society last year. There's a nice poster on the NS Pharma website that you can see some of that data. So not the parent company website, but the actual NS Pharma website. And we did look at, as you probably know, time to stand, which was, we compared it to historical data and control, and at 24 weeks, that was statistically significantly different in favor for the 40 and 80 milligram patients. We looked at 6-minute walk-run test and several others of the functional tests, most of which were statistically significant based on comparison to historical controls from the Synergy cohort. So that data, if you go on our website, that data, you should be able to find it. And we're in the process of putting that data together for a manuscript, which we hope will be published within the next several months.
Okay, thank you.
One more question here, it looks like.
Hi, this is for Sarepta. I'm asking this for parents watching from home regarding your preclinical products that have been in preclinical for a long time. They're asking specifically the SRP 5050 product, when it might get out of preclinical, like a realistic timeline for all of those products.
Yeah, that is the question I hoped to answer in our slide. What we've done is we're actually doubling down on the 50, so just to be clear, the 5050 is a PPMO. This is the peptide PMO. And so we have actually doubled down our effort on 5051 to essentially use the data that we get from that to make critical decisions. And what we're actually, our trial is designed to do is to say, prove that, or test the hypothesis that the conjugated peptide does indeed enhance PMO delivery. Once we actually have that proof, we will be looking at those data and in the context of where we are with PMO and gene therapy, making that decision in mid-2020.
Could it potentially roll right over into PPMO, the 5050, some of those that are in preclinical, and skip the PMO?
So I think that that's the kind of decision we actually will be making, because when you have chemistries, when you have a number of different platforms of chemistries, what we want to do is actually use that 5051 data set to actually help us decide, okay, have we genuinely enhanced, and then what do we do? Does that make sense?
Yep.
Yep.
I think that'll help the folks watching from home. Thanks.
Thank you.
It looks like one more question.
Thank you. My name is Laura McLinn. My son is also one of the 16 boys in the NS Pharma trial. And my question is about the next phase. Will that include non-ambulatory? And what dosage will you choose for that?
For the phase 3 study, again, it's, that one is still in negotiations with the FDA. They have to discuss it with us and give approval as part of the NDA process that we're doing on the phase 2 data. I don't believe those that study will have non-ambulatory. However, we are looking, and we recognize the need for that type of information. So we're also looking at other studies as well, and not just this one phase 3 study, but a suite of studies that can then be a little bit more flexible in terms of age and state. The Japanese study did contain 3 patients who were non-ambulatory, so we're going to focus in on some of that data and take a look at it. So we know Viltolarsen has been given to patients in Japan, 3 of them that were not ambulatory.
So just to make sure I understood that, you're not sure if the phase 3 will be the 40 milligram or 80 milligram dosing, right? Is that what?
No.
You're waiting to hear from the FDA on that?
I'm assuming it's going to be both, but I don't want to say the final dose until we have our discussions. I'm pretty certain it will not have ambulatory patients.
Mm-hmm.
Well, it will have ambulatory patients in it, but that doesn't preclude us doing other additional studies outside that phase 3 study to explore other inclusion criteria. So we don't want to just have one more study going on. We want to have a suite of studies going on.
Thank you very much, and thank you so much for keeping our boys on drug.
And thank you. The 4 presenters were great, great presentations. Everybody stayed on time or beat the clock. And I think Ryan has polling questions to keep everybody excited. Thank you.
Okay, this is the last time I'm going to bug you all, so please stay. Sounds so desperate, don't I? Please. So we're going to have 5 questions, but this will close out the polling. I appreciate everyone that's participated. It's been really helpful to inform the discussions throughout the week. Okay, you could pull up the first one. Since we've been talking so much about clinical trials, we want to understand if you were deciding to join a new trial in the next few months, which 3 things would make you most likely to join that trial? Lots of votes for. Seeing a lot of trends from last year where we asked this question as well. We wanted to repeat it this year, see if there was some comparison. Access to the drug and placebo pieces are once again high on people's lists. Okay. Looks like most votes are in. We can go to the next one. So on the other side, what 3 things would make you least likely to join a trial? You can choose 3. Very similar trends to last year's. So we're seeing some consistency. Some votes are still going. I'll give you one more minute. Hopefully everyone can get to the website easily. Okay. Let's go to the next. This is a word cloud. Since we had CRISPR this morning, what word comes to mind when you hear the word CRISPR? It is adorable, I know. I named my cat CRISPR. Coming. We have 2 more questions after this. I really thank you for holding on. We see hope, cure, potential. So like cute. Okay, I want to move on to the last 2 questions. I want to understand over the past 2 years, how often has your or your child's doctor talked to you about opportunities for trials and advances? High numbers for very often so far. Coming in, I'll give you 30 more seconds to get on there. Looks like the bulk are coming in still. Okay, and the final question, we're very interested to know, has your physician spoken with you about clinical trial options outside the clinic you currently go to? So they may not be running that trial at the clinic you attend, but are they talking to you about trials outside of that specific clinic you attend? It's very informative. Almost 50-50 there. Very interesting. Okay, I want to thank everybody for doing the polling.